systematics. So the phylogenetic approach to classification is a cornerstone of a branch of biology called systematic taxonomy or systematics. This organizes the monstrous diversity of living things in the context of evolution. They use several lines of evidence to construct a phylogenetic tree. So a phylogenetic tree, which you see here in the picture, is a family tree that shows evolutionary relationships that are thought to exist among groups of organisms. So you can see, let me get my laser pointer. From the phylogenetic tree that you see there, basically what you're looking at is kind of like the evolution of life, starting with the chemical origin of life, which I talked about like way back in the beginning of online learning when we looked at how the precursors of organic molecules came into existence, and how you had like the coacervates and the microspheres, and you had like organic compounds that were made. And then you've got your Monera. This is kingdom Monera. These are the eubacteria and archaebacteria. So those were the first organisms to evolve. And then from there you had the protists. And of course, then you've got more and more branches as life started to evolve on Earth. The tree represents a hypothesis based on several types of evidence. So the types of evidence that we use to try to form a relationship between the organisms are the fossil record, morphology, the embryological patterns of development, and chromosomes and macromolecules. Remembering that morphology means the structure, the internal, external structure. So we're going to start with the fossil record. This provides clues to evolutionary relationships. I've talked about it before. When you look at the fossil record in a particular geographical area and you compare it to extant or currently living species, you guys all know what extinct means, right? Like they don't exist anymore. The opposite of extinct is extant. Just might be like a SAT word. Extant is the opposite of extinct. So these are currently living organisms. Some organisms have complete fossil records, but to be honest, most of the fossil records are incomplete, but it provides a framework for the phylogenetic tree. And I don't know what confirmation is incomplete, probably, I don't know. The second one is morphology. So we can compare the structures and the physical appearance of organisms. My earphone's getting caught. Right, so part of the evidence of evolution that I talked about in a previous chapter are homologous features. Homologous features are an indication that two organisms descended from a common ancestor. But we have to be sure that the homologous features are true homologous features and not really analogous features. So, for example, insects and mammals both have legs, but that's not a homologous feature. Just like the wings of an insect and the wings of a bird, those are analogous. Even though they look similar in structure and they have the same function, they do not have the same the same genetic origin. So analogous features are not an indication of ancestry, but homologous features are. The next evidence that we look at to establish evolutionary relationships is embryological patterns of development. And I've talked about this before. You've got here the embryo of an amphibian, reptile, bird, and mammal, and you see just how similar they are as an embryo, but you can look at the embryo of, say, an invertebrate, because these are all vertebrates. And if you remember kingdom, phylum, class, order, family, genus, species, we have kingdom, Animalia, and phylum, Chordata. These are where all of the vertebrate animals are, all the fish, amphibians, reptiles, birds, and mammals. So all of the chordates share a common ancestor insofar as that we all have a backbone, and you can look at the embryos of the chordates and you can see that they have a similar embryological pattern of development. We all develop as embryos in a similar way. As development begins, the zygote divides by mitosis. Within hours, a ball of cell called a blastula forms, and a small indentation called a blastopore develops on the outside of the blastula. This is how vertebrate embryos develop. The blastopore eventually becomes the digestive system. Now, in most animal phyla, the blastopore becomes the anterior or the front end, the mouth end. But in echinoderms are like starfish and sea urchins, it becomes the posterior or the back end, just like chordates, like us, animals with a backbone. So this is why it's really important to look at how embryos develop, because echinoderms, like starfish, obviously, these are invertebrates. And the chordates are the vertebrates. But if you look at the embryo development of a starfish, the blastopore forms the anus first, just like all of the vertebrates do. So they're actually more closely related to us than they are to other invertebrates like jellyfish or squid or things like that. So that's why it's important to look at many different pieces of evidence and not just saying, well, they're invertebrates, so they must be more closely related to other invertebrates, because it's in fact not true. Starfish are more closely related to the vertebrate animals than they are to other invertebrates. Another similarity between the echinoderms and the vertebrates is in their stem cells. If you don't know, stem cells are the original cells that form shortly after fertilization. And these are undifferentiated cells, which means they have the potential to become any type of cell. They can become a hair cell, a heart cell, liver cell, skin cell, whatever. In early development in both phyla, so this is both echinoderm and vertebrates, any cell is capable of forming the entire organism. For identical twins, you have one egg that splits into two. Other animal phyla do not have this. For example, if you split a lobster egg in half, one half forms one part and the other half forms another part. And both parts obviously are going to die because they're not forming the full organism. So it's interesting if you think about what we understand about human biology, that a lot of times we apply that to all of the animals or all life, actually. So if you say, okay, well, if humans have the ability to fertilize an egg, and then if that egg splits in half, it forms two completely new individuals. So that must be true just as a principle of biology. That's actually not true. So the echinoderms, which is the phyla that, like I said, that includes the starfish and sea urchins and animals like that, they have the same ability that if they have a fertilized egg and you cut it in half, it can form two completely new individuals, just like vertebrate animals. But other animal phyla, like I said, like if you look at an octopus, a squid, a snail, what else? A sponge, lobsters, crabs, whatever, they don't have the same characteristic, which is what puts echinoderms more closely related to the vertebrates than other invertebrates. And the last category that we look at to develop a phylogenetic tree are chromosomes and macromolecules. So the chromosomes, obviously, we know what those are. The macromolecules are going to be DNA, RNA, and proteins, and we use them as a kind of molecular clock. The number of amino acid differences is a clue to how long ago two species diverged from a shared evolutionary ancestor. So again, when you look at a phylogenetic tree, and they're usually shaped something like that, and you get branches that come off, whatever. When you are looking at two organisms, let's say this is organism number one, organism number two, Because a lot of people don't understand how evolution works. And I don't know if I was talking about this with you guys or a different class, but I see so many times, especially when you get on social media where like the stupidity knows no limits, that people say, well, if humans evolved from apes, then why do we still have apes? Like, wouldn't they all be gone? And the problem is, it's like, we didn't evolve from apes. That's the number one, like, obviously, they don't show an understanding of how evolution works. How evolution works is that we are related to the apes and we share a common ancestor with the apes. So if you see one of these branches is humans and the other branch are the apes, and you go back in time, which is when you go back down the line of evolution, and you see that we have a common ancestor. And I've talked about in other chapters about how new species are formed. So at some point in the past, you had a common ancestor of both human and apes. So humans did not exist, apes did not exist. You had this common ancestor, and this common ancestor split off and formed two different lines of descendants. One of those lines of descendants would eventually become humans, and the other line of descendants would become apes. And that's why there are still apes in existence today, because we didn't come from apes. Okay, so we look at the sequence of amino acids. So basically, the more similar the sequence is, the more closely related and the more recent the common ancestor between those two species. The more different the amino acid sequence is, then you have to look further back to find a shared ancestor. Of course, it's not a perfect clock. This method assumes that all of the changes in amino acid sequences are random and not affected by natural selection. But of course, that's probably not true because natural selection is always at work. You also have changes that occur at different rates in different species. But if you put together the macromolecule amino acid sequence together with morphology, with a fossil record, with embryological development, you put them all together to form a hypothesis about the evolutionary relationship between organisms. Now, we can also compare chromosomes. We can compare the karyotypes. The karyotype is a photograph of the entire set of chromosomes of an organism. And you can have areas of the chromosomes that have the same pattern of banding, and that can be a clue to the relationship between species. For example, chimpanzees and humans. Chimpanzees are our closest living relative. I think the DNA of a chimpanzee and a human is 98, so around 98% identical. Identical means exactly the same. So if you think about a chimpanzee and how different we are, that is only 2% difference in our DNA. That's how closely related we are. One chromosome in humans is actually homologous to two smaller chimpanzee chromosomes. So this is a karyotype, and you can see the pattern of banding, the patterns of banding on the chromosome, and you can compare chromosome sets between two organisms, and you can see how some of the banding is similar. And that will also show you relationship as well. So I'm looking here at the X chromosome, and you can see the similarity between the human and the chimpanzee. All the banding looks pretty identical, actually. A little bit different at the end here. So you can compare karyotypes as well. Now, the last thing is cladistics. It's a relatively new system of classification, and cladistics uses what's called derived characters to establish evolutionary relationships. Derived characters are features that apparently evolved only within the group under consideration. So it looks at a characteristic or a feature that a group of organisms have in common that they don't have in common with other organisms. For example, in birds, feathers are a derived character. Feathers are a feature that all birds have, but no other organism except birds have. So it's safe to assume that feathers evolved within the bird group, and they were not inherited by some distant relative. Although we do have evidence now that some of the species of dinosaurs had feathers, which is one of the biggest pieces of evidence that we have for that, the fact that birds evolved from dinosaurs. Cladistic taxonomists say that organisms that share derived characters share it because they inherited it from a common ancestor. So this is really strong evidence for common ancestry. And if you think about it, it makes sense. If all birds have feathers and no other organisms have feathers, it makes sense that they inherited feathers from a commonly shared ancestor. Cladistic relationships are shown in diagrams called cladograms. So this is an example of a cladogram, and each of these characteristics here is what separates everything from above that characteristic, separates them from the organisms below. So for example, you start with a vertebral column, so that would separate it this way. So the lancelet doesn't have a vertebral column, but all of the other species that you see above that characteristic do share that characteristic. Then you have the evolution of jaws. Lamprey do not have jaws, so that separates all of these guys from the lamprey. Then you've got 4 walking legs, and that separates the salamander, turtle, and leopard from the tuna. Then you've got an amniotic egg, and that separates the turtle and leopard from all of the other ones. And then finally, hair is only shared by the leopard. So it eliminates species or groups of organisms that don't share that particular characteristic from everything else that does.